Alright, so when we're looking at this slide over here, this one here, first of all, can you tell what organ it's from? Okay, small intestine, and what specific portion of the small intestine? The ileum. Okay, good. And how would you be able to tell this? Okay, the Peyer's patches. Very good. Okay, if you were to zoom up on one of these things here, what is this structure that you have here? This is a villus. Yeah, a villus. A circular fold, if you do see it one, okay, it will be like extension of this, okay, and then you have the villus coming off of it. Okay, and to also distinguish it, if you look at this, right, these are it's not the microvilli, right, it's the simple columnar epithelium, right? On the surface of this, which it's not that zoomed up, then you could see the microvilli or the brush border. Why do we have a brush border or microvilli? Increased surface area for what? Especially when we're talking about the small intestine. Absorption, yeah, of course, there's going to be digestive enzymes on the microvilli area as well, and those digestive enzymes will help with breaking down the biomolecules into smaller particles. Okay, good. And if we're talking about what's inside here, what is inside here? Okay, it's part of the mucosa, right? What's underneath the epithelium? Okay, good. Connective tissue. What is that connective tissue with the other tissues involved at this location, though? Starts with a L. Yeah, the lamina propria, right? Good. And what would you call this here? This smooth muscle that you see over here? Yeah, the muscularis mucosae. Okay, good. And then so all collectively you have all these parts making the mucosa. Very good. What do you call these dips that goes down here? That you see a little bit over here as well. Okay, it's not pits because we're in the... What organ are we in again? Okay, this is the intestine, right? So... No, the goblet cells you'll see within the columnar epithelium. So these lightly stained or non-staining structures that you see here are the goblet cells. Not the circular folds. Circular folds, once again, are extension of the muscularis mucosae, where there's multiple villi that comes off of that. You don't see any circular folds on this one.
So the villi come off just from the circular folds, or they can...
Yes, so you have to look at the... Here, let me go back. I think this is... No, this doesn't show you. I think when we looked at it, it was over here? No.
That wouldn't be the blood vessel itself, would it?
Sorry?
Would they be the blood vessel?
No. Okay, so it wasn't there. Where was it? I thought I had it. Oh, here we go. Okay. Notice how, okay, same structures, right, that we were looking at, right? What was the one of this? Villus, very good. And notice how these villi extend off of this structure here. So collectively, this thing is a circular fold. And then if we look at the epithelium here, right, they have the microvilli on the cells, right, the simple columnar cells. Do you see the difference now?
So the empty holes there, the white
Here?
circle, yeah, and also pink circle, those are empty the blood vessel or not?
Yeah, correct, And that might have some blood vessels going through in it. Exactly. All right, so, but looking back over here, when we're looking at this, what are these things that dip downwards here? Yeah, intestinal glands. Intestinal glands. Okay. And then when we're looking at this layer down here, what layer are we in now? Submucosa, right? And these structures are the Peyer's patches, right? Pull the malt, right? The mucosa associated lymphatic tissue. And then underneath you have this layer here. Not adventitia. It's a muscle layer, right? Not the serosa. Yeah, it contains this inner circular layer, right? This is the inner circular layer and the outer. No, the muscular mucosa, we talked about it earlier, right? It's near the mucosa layer here. Muscularis externa. Okay, and then the layer underneath the inner circular layer, it's the outer longitudinal layer. And if we were to look at the surface of this here, then we have the, in this case it's serosa, because serosa is just the simple squamous epithelium with a small layer of areolar connective tissue underneath that. Okay. All right. Just trying to get you guys more associated with these histology, since you'll be seeing a lot of the histology for this particular chapter, this unit especially. Okay, so make sure you pay attention to that. Okay, so now going into the large intestine, since we finished the small intestine. Small intestine is actually really long. The reason why this is called the large intestine is not how long it is, because it's actually shorter than the small intestine. However, diameter-wise, it is larger. Okay. And at this location, remember how we talked about chyme? What is chyme again? It was what? No, that's chyle. Yeah, That's chyle. Chyme. Okay, bolus was what? Food plus saliva, right? Chyme is whatever was the bolus, right? Plus what? Okay, gastric enzymes and juices. And at this point, it's also intestinal juices already involved too, as well, right? Okay, that includes pancreatic juices and also bile as well, and so on and so forth. Okay, so all this, once it gets into the large intestines, instead of calling it chyme now, we're calling it feces. Because most of the absorption is done. Most of it is done, so it's like more waste products at this point. Okay. So its function, one, is that there is peristalsis. Once again, peristalsis is not technically a mechanical digestion. What is peristalsis again? It's just, moving proportion of the feces in this. It absorbs some absorption, but very minimal. But if it does absorb stuff, it's absorbing water, electrolytes, and vitamins. So it doesn't absorb like the any more glucose, amino acids, lipids at this point. All that was done in the small intestine. Okay, so if when it does absorption, it absorbs these things here, and it stores the feces. And okay. It stores the feces as well. And so that we could potentially go number 2 when we want to go number 2. Okay. And this is also done by having this both the inner anal sphincter and also the outer anal sphincter to be contracted until it gets full. Once it gets full, then the smooth muscles, the inner anal sphincters, tries to relax. However, the external one, which is the one that we have skeletal control over, we tend to want to contract it until we are ready to go, basically. There are different regions in this large intestine. What was the different regions in the small intestine? Duodenum, jejunum, and ileum. Very good. For the large intestine, we first start off with the cecum, right? We start off with the cecum. What was this little opening from the ileum to the cecum called? Starts with the I. Ileocecal valve. Ileocecal valve. So with this cecum, coming off of it is the appendix called the veriform appendix. This veriform appendix is actually useful in some sense. Like it actually contains a lot of lymphatic nodules in here. So meaning that it actually helps combat against like bacteria and any type of infections that might be harmful to us. And usually it'll come out of the appendix here and go into the large intestine. Okay, of course, some people has this inflamed, and when it's inflamed, potentially it might burst, causing the exposure of a lot of the bacteria within the large intestine to come out and invade the peritoneum, which we don't want, and that's hence why they would extract the appendix out of there.
But why does large intestine have those like fats like hanging out when all the other organs within the abdomen doesn't have fats?
There will be fats pretty much even within the surrounding our organs too. Like remember the heart? We saw a lot of fat around the heart. So there is a lot of fat around the tissues. So it's kind of serves a couple purposes. One is to cushion the whatever organ that it's covering. Another is potential, fats are for what? Generally speaking, fats for a source of energy as well, right? So a few of those reasons. So yeah. And when we're talking about the colon, this is now the region between the ileocecal junction and the rectum. So we have the one that goes upwards this way, known as the ascending colon. And this corner over here, when it becomes the ascending to the transverse, this is called the right colic flexure, or you could call it the hepatic flexure, since the liver will be kind of around there. Okay, and then you have the transverse colon here, going horizontally here. Here's the transverse colon. And then it comes here at this corner over here. And this corner is going to be called the left colic flexure, also known as the splenic flexure, because the spleen is located around here. And what would you think this part will be calling when it goes down this direction? Yeah, descending colon. Very good. This descending colon. And then there's this S-shape part here. This S-shaped part is going to be called the sigmoid colon. And then we get to the rectum. And the rectum is this area. And what was the function of the rectum again? To store the feces. Store the feces. There's an anal canal over here on either side. This anal canal passes the feces during defecation. And of course, we have an internal anal sphincter, and the internal anal sphincter is once again a smooth muscle. It's contracted unless they feel pressure at this area. They feel pressure because of the feces that's being stored there, right? And so once they feel pressure, they're going to relax. However, we have an external anal sphincter, and this is the voluntary skeletal muscles. So when we feel pressure, that means okay, the smooth muscle is starting to relax at times, and then we want to feel like we have to go, but we still have our external anal sphincter to contract to prevent us from going until we want to go, kind of thing. Meaning like, hopefully you're sitting on a toilet and stuff like that, and then you relax the external anal sphincter. So the smooth anal sphincters, it will relax when it's filled, but depending on if we're doing our external anal sphincter, we're contracting it, there will be a less action potential sending it to the smooth anal sphincter, but then they come in waves. Okay, so. Alright, the large intestines, we have the intestinal glands or the intestinal crypts. And once again, these are these structures that goes downwards. And you'll notice that there's no villus over here. Okay, so the small intestine, we notice that the villi goes up, and then there's parts that goes down like this. Right. For the large intestine, things just go downwards like this. Okay. So there are no villi. The mucosa layer once again has it's not smooth, but simple columnar epithelium. Once again, it's to absorb water, electrolytes, and vitamins. They do contain a lot of goblet cells, like a lot. And partially because since we're absorbing water here, right? The feces are going to be pretty solidified, like meaning that it's not going to be as liquidy, right? Because we're absorbing the water. So it's going to be drier and drier and drier. And being drier and drier and drier, it's going to cause more abrasion potentially against the large intestine. And hence why we have a lot of goblet cells to produce what? Goblet cells produce mucin, mixes with water to make mucus, right? And that helps lubricate the feces, the dried out feces, as it's passing through the large intestine. Then we have the muscularis over here, which is just the outer longitudinal layer. Okay, the outer longitudinal layer is complete. Right, the muscularis externa. And then this muscularis externa is just the outer longitudinal layer. There is the tinea coli, right? Okay. And then this one is made up of smooth muscles that causes the haustra. So one haustrum is one of these structures that you see here. Okay, so these are multiple haustra. And what causes it to form their shape over here are smooth muscles that form these haustrum. Okay. Okay, so regarding to the muscularis externa once again, this layer is the inner circular layer. The outer longitudinal layer is not complete. The outer longitudinal layer is just at this one part here. So when you're looking at the histology, you might only see, potentially, you might only see only one layer at the muscularis externa. Just depends on how they cut it, right? If they cut it, just say over here, right? They just cut it and took this slice over here at this area, then they don't have it. But if they cut it anywhere along this plane here, then okay, they might have a little bit of the outer longitudinal layer. Okay, once again, we have the haustra. And the tinea coli is these smooth muscles that you see over here. That causes the haustra. At the tinea coli, you see these fats that come off of it. This is called the epiploic appendages, also called the omental appendices. And these are just small fats that hang on the tinea coli.
The intestinal gland, they go into the submucosa. So is the diagram
The diagram's not incorrect. So it's going towards the submucosa, going downwards kind of thing. So it's going just downwards. That's what I'm basically trying to imply. Versus the villi, right? They go upwards. Does that make sense?
Yeah, just so I know from
Mhm.
histology, like, so I know I can distinguish the layers.
Mm-hmm. Yeah.
Are the epicolic appendages the same as the omental?
Mm-hmm. Yeah. Yeah. They're the same thing. Just depends on which textbook that you're looking at. They call it differently, but they're the same thing. Yeah. Okay, so here is the large intestine. And once again, if you look at it, the surface here tends to be pretty uniform, more or less, right? But you see these indentations that goes downwards. So one of these structures that goes downwards like this, these are the intestinal glands, right? Once again, if we look at the surface of this, we have what kind of tissue that we have here? Simple columnar epithelium, very good. And notice how there's a lot of what? Goblet cells here, okay? So these are things that you want to clue you in and to figure out, oh, this is the large intestine. First, there's no villi, right? Second, if you look at it, there's a lot of goblet cells, okay? Once you see a lot of goblet cells, not to say, the small intestines, they do have some goblet cells, but it's not as prevalent when you compare it to the large intestine. Okay, good. And then if you were to say, you don't see much of it here, though. Okay, but what's underneath the epithelium? Here? Or here, that you see? What's underneath the epithelium? We just went over this, not the muscularis, lamina propria. Very good. Okay, and then you have this smooth muscle layer here. That's the. Say again? Not the submucosa yet, right? Let me take this off so you can see the division, right? You have the division somewhere around here, right? So what is this here? Say again? Yeah, muscularis mucosa. Muscularis mucosae. Okay, so making this the mucosa layer. Okay, good. And what is this layer here? That's the submucosa, and then we do see two over here because it did cut right in the middle here. So what is this layer here when we look at it fully? Muscularis externa. Muscularis externa, having the first here layer is the inner circular layer and the outer longitudinal layer. And then the very outer surface here, the serosa, once again. Okay, if it's adventitia, okay, and there will be adventitia like near the rectum and anal canal and stuff like that. It will be just connective tissue that you'll see all over the place. It wouldn't just be that one layer.
Where would the lamina propria be?
If you look at the lamina propria, it's underneath the epithelium. So all this that's here
Okay.
is lamina propria. Oops, sorry, I didn't mean to do that.
So it doesn't have the goblet cells like in the picture. It would just be empty of that.
Oh, there's all these structures that you see that you don't see it stained over here. These are all goblet cells.
No, inside of the lamina propria. No,
Epithelium.
just
Yeah, just the epithelium. Okay, let's just go into the questions here. Okay, so that was all part of the alimentary canal, right? Alimentary canal is what again? It's the entire digestive tract from your oral cavity all the way to your anus, right, kind of thing. Okay, so next we're going to be talking about the liver is part of what part of the digestive system again? Accessory, accessory organs of the digestive system. All right, so its function is to produce bile, okay? And bile is for emulsification. And that's a fancy word of helping breaking down the fats into smaller fats. Remember how we were talking about like how our teeth helps with breaking down food particles into smaller food particles so that the digestive enzymes has more surface area to help digest the smaller food particles. This is kind of like the same thing that's it's trying to do right now, okay? So technically, it's not, it is, okay, in some sense it's breaking certain bonds into smaller fat molecules. So it's not breaking down the fats into the particles that make up the fats, okay? What I'm trying to get at, a triglyceride, which is fat, right? Another word for fat. It's made of something called glycerol molecule plus 3, hence the fatty acids. Okay. In order to break down a triglyceride into glycerol and its 3 fatty acids, we need lipases, okay, which is the enzyme that breaks them down. Okay. So in order for triglyceride to become glycerol and 3 fatty acids, we need lipase. However, bile is not a lipase, okay? It just helps emulsify the fats, okay? So you could think of emulsification kind of like, okay, if you were to leave a salad dressing, Italian dressing on its own, right? You have that layer of oil on top, right? If you were to shake it up and stuff, you notice that that layer of oil become droplets of oil instead, just temporarily, you know. But what bile does, it makes the fats into more like those droplets and keeps it in those droplets so that, once again, you increase the surface area so that the lipase can break them down into the glycerol and fatty acids. Okay, do you understand the differences now between what bile does and what the lipases do? Okay, bile just makes them into smaller fat droplets or fat particles so that the lipases has more surface area to break the chemical bonds of triglycerides into fatty acids and glycerol. Okay, another function of the liver, it helps detoxify drugs. And it helps synthesize certain blood plasma proteins, and especially which blood plasma proteins we talked about? Albumin, yeah. Mm-hmm.
And also fibrinogen.
Yeah, fibrinogen. Yeah. Mm-hmm. And it helps break down damaged cells like erythrocytes. And we talked about how the liver is also a storage, right, of glycogen, right? And so when our blood glucose gets too low, we have glycogen being broken down in the liver so that it releases glucose in the blood. So you could say breakdowns glycogen, or you could say stores glycogen, right? Same thing. Same thing, but it stores it up so that it can break it down. Yeah. Okay, the liver has different lobes. We have the right lobe, which is this large lobe over here, a left lobe on the other side. And we take a look at the inferior side here. Then you'll notice that there's two other lobes here. There's this one here, and there's this one here. Caudate, whenever we think of caudal, we think of what? Towards in which direction? Yeah, towards the tail. Okay, good. If we're looking at, like, say, a cat or something like that, it's towards the posterior, right? So you could think of that when you're looking at the liver. Okay, so what is this vessel here? Can you guys tell? Inferior vena cava. Okay, so right next to the inferior vena cava, you have that caudate lobe. Okay, just anterior to that caudate lobe, you have the quadrate lobe. Okay, the quadrate lobe is right next to the gallbladder. So when you orientate this, then it helps you distinguish, next to the inferior vena cava, caudate lobe. Next to the gallbladder, quadrate lobe. There are the different ligaments, okay? The falciform ligament is this ligament here. And this helps secures the liver to the anterior abdominal wall. There's also a round ligament, and this one we've talked about before, right? The round ligament is the structure that comes off here. And what was it used to be a part of? The umbilical vein, right? And then we have the ligamentum venosum. And that used to be what? Ductus venosus. Very good. And that used to go into the inferior vena cava. Okay. So these structures are remnants of the fetal circulation. There's something called the porta hepatis. And this is an opening between the quadrate lobe and the caudate lobe. Okay, and if you look at this, it's just this opening over here. And this opening allows these blood vessels to go through, and also the common bile duct over here. So here, if you look at it, you have the hepatic portal vein. You have the hepatic artery proper. And you have the common hepatic duct here. So some of these are terms that it's just to review, things that you should already know from the last unit. And here's a new terminology here, where it's called the common hepatic duct. Okay. On the liver itself, there's this area called the Baer area. Okay, most of it is on the right lobe over here, and it's just this part where it's not covered with peritoneum. It's directly in contact with the diaphragm. And you'll see this on the model. It's kind of colored a little bit differently compared to the rest of the model. The liver has these hepatic lobules, and some of these you should know. Not all of these, but we have hepatocytes, which make up most of the hepatic lobule, and these are the liver cells. These liver cells, they make bile, they help detoxify the blood, and any kind of nutrients that we have too much of, it stores up. And it also makes those plasma proteins that we just talked about. So all those functions that we talked about regarding to the liver. So these are, once again, what kind of cells? Hepatocytes, okay. What was this structure called again? The hepatic portal vein, or the venules, right? And this passageway, sinusoids to the central vein. So some of these you should already know, right? Okay. When we're talking about these areas in between the hepatic lobules, these are called the portal triads. It's called the portal triad is because of the hepatic portal venule that goes through it. And also because you also find at that location a hepatic artery proper. We have the hepatic portal venule here, and also something called a bile duct here. The bile ducts are these drainage area for the bile canaliculi. Okay, these are the bile canaliculi. So the hepatocytes, they make the bile. They release the bile into a bile canaliculi or canaliculus, and it goes to the bile duct over here. We already know what the hepatic sinusoids are. These are the capillaries, and once again, that's where it gets filtered. Not really filtered, but where it gets detoxified, the blood and whatnot, and have albumin being released into these areas and stuff. Inside these hepatic sinusoids are these cells called the Kupffer cells, or the reticulo endothelial cells. And these are phagocytic cells that once again help detoxify our blood as it goes into the liver. Central vein is where the hepatic sinusoids drain to. And once the central veins collectively, they'll get, into the inferior vena cava via the hepatic veins here. The bile canaliculi, like we just mentioned, drains the bile from the hepatocytes into the, what do you call this structure here again on the side? The bile duct, So if you were to take a look at this on a slide, okay, so once again, one of these structures here is a lobule, hepatic lobule, okay, hepatic lobule here. If you look at the hepatic lobule, you'll notice this central location here. That's the central vein. Very good. And then you see the spaces here. What are those spaces called? Sinusoids. What do you call all the cells that you see here? Hepatocytes. Very good. Very good. If you look at the in between the hepatic lobules, you have the portal triad. Okay? And when you look at the portal triad, I don't have a slide for it, but take a look at the hepatic triad. What you'll see is, first of all, you'll see a hepatic artery proper, right? If it goes in here, you have the hepatic artery proper. You also have a hepatic portal vein, right? So remember how we had to distinguish what's an artery and what's a vein? You'll see that again occurring over here. Okay, so you'll need to be able to distinguish, what's the hepatic artery proper, what's the hepatic portal venule. Okay, and then so those two you should be able to distinguish. The one structure that you haven't seen before is the bile duct over here. The bile duct, if you look at it, it'll have these like simple cuboidal cells that basically make up a duct. Okay, so it looks something like this when you look at under the microscope. Okay, so this is like a bile duct here. And they're pretty obvious when you look at it in comparison. So even here, when I look at it, this structure that I see over here, I could tell that's the bile duct right there. Okay, of course you have to zoom in a little bit closer to see the details of it, but take a look at it under the microscope.
Does every lobule have only one bile duct?
No. It'll be multiple. It just depends. Like over here, there might be another portal triad over here. Okay, so here's a portal triad over here. Once again, the canaliculites, the bile canaliculites, will drain to the bile ducts, basically. So blood flow to the liver, we have blood coming in from the hepatic portal vein and also the hepatic artery proper. They go into the hepatic sinusoids, going into the central vein. And from the central veins, they'll collect it into the hepatic veins here, into the inferior vena cava. So this should be a review, at least, right? This part. The gallbladder, its function. Okay, what makes bile again? The hepatocytes in the liver, okay? The gallbladder, it stores and concentrates that bile that was produced from the liver. And does this by going through the cystic duct over here. So what bile acid and salts are? These are steroids from cholesterol, byproducts from cholesterol. And once again, it helps with the digestion of fat. How does it help with the digestion of fat again? Bile. How does bile help? Emulsification, right? Helps breaks the fats into smaller droplets of fats so that we increase the surface area so that the lipases could digest the fats into glycerol and triglycerides. Glycerol and fatty acids. Good question. Okay, so if you cut it off, what do you think will occur? Okay, not that it's no bile, right? Because where does bile come from? The liver. Okay, so they'll still be able to produce bile, but and they won't have an excess amount of bile, right? What's the function of the gallbladder? To store and concentrate the bile, right? Okay. If it's storing and concentrating the bile and you don't have this structure to help store it and concentrate it, okay, you still have some bile. What do you think you need to do diet-wise to help you in this aspect since you don't have a gallbladder? Yeah, you cannot eat as many fatty foods, okay, or oily foods. Okay, because you don't have the amounts of biles to help break down the oils in those foods, in those fats in those foods. Does that make sense?
Yeah. But then does the fat actually get absorbed into the body, or it just kind of passes through?
It should just pass through, because if you think about it, if it's not able to break it down, right, then it won't be able to be absorbed as much, right? But you do need, technically, our body actually produces like fats themselves, so technically we don't have to consume it, but we do need some in some aspects, because one, it provides energy, right, and two, like certain like our cell membranes and cholesterol and stuff like that are necessary to form our cell membranes and different hormones and stuff like that. So we do need some. Our body does produce some to and converts glucose or amino acids into fats. So yeah.
People will still get fat even though they don't have gallbladder, even though they cannot eat fat.
Yeah. So yeah. They could. I mean, once again, it depends on what they are eating too. Like if they eat too much sugars or too much carbohydrates, too much amino acids and stuff like that, they're going to still store it up as fats, because your body converts it from carbohydrates into fats, from proteins into fats and stuff like that. So once again, it's everything in moderation in that aspect. So regardless, just don't eat too much food, basically. That's the lesson. Yeah. The gallbladder looks like this structure here. You don't actually have to distinguish any parts to this, okay? Just be able to recognize this. And this is different from all the different organs that we've seen thus far. It tends to be very branchy kind of thing. So when you look at something like this under the slide, you should be able to distinguish that this is the gallbladder. Okay. Once again, we're not looking at any of the specific structures inside of it. Just recognize it. If you see this and it says ID the organ, that'll be the only thing I'll be asking you, is to identify the organ. Okay, there's this thing called the biliary apparatus. This biliary apparatus allows bile to flow through all these different structures. Once again, bile is produced by the hepatocytes. Very good. And that's where we start over here. The hepatocytes, they produce the bile. The bile will go into the small vessels called the canaliculi, right? The little passageways called the canaliculi. From the canaliculi, it will collect into the bile duct. Okay, so those parts we already talked about. Okay, so here's the bile canaliculi, bile duct. Okay, and from the bile duct, this is now going to be collecting it from the hepatic bile ducts, both right and left hepatic bile ducts. Okay, this is right, this is left hepatic bile ducts. The right and left bile ducts will connect together and make the common hepatic duct here. The right and left bile duct will fuse together, making the common hepatic duct. And remember how we were talking about the structures that we see out of the liver here? When we looked at the liver over here. Remember what was this again? The hepatic artery proper. This one is called the hepatic portal vein. This that's coming out of it is this common hepatic duct. Okay, so those structures, those terms you already should know. Okay, and what ends up happening is that when you look at what was this again? Hepatic portal vein. This one, hepatic artery proper, and then this one here. Oh, sorry, my bad. Let me take this off. Okay, these are all coming off the liver, right? So this is, if you look at this one here, this is coming from this lobe over here. So that's the left hepatic duct here, and this one here is coming from the right lobe. That's the right hepatic duct. And now that they fuse over here, this is called the common hepatic duct. Okay, and we said that where is bile stored at? Gallbladder, right? Okay, so we have to go to the gallbladder. Okay, so this common hepatic duct will go to the gallbladder through this passageway here. This passageway is called the cystic duct. Okay, and it's stored in the gallbladder. So when we start to eat fatty foods or oily foods, bile from the gallbladder will start to come from the gallbladder through, what is that structure called again? Yeah, it will go through the cystic duct and continue onwards, okay? So from here, this is called the common bile duct here. Okay, so from the cystic duct and where the common hepatic duct goes to is the common bile duct here. And from the common bile duct, then, if you notice here, same structures, right? The right and left hepatic ducts, the common hepatic duct, cystic duct, going back and forth. Here's the common bile duct. Common bile duct will fuse together, okay, with this major duodenal papilla. This is from the main pancreatic duct here. They will end up forming the major duodenal papilla here. Okay, and they will end up going to the duodenum. So once again, common bile duct fuses with the major pancreatic duct here. They go to this area which is a little bit more enlarged, called the hepatic pancreatic ampulla. Go out of the duodenal papilla here and out into the duodenum. Okay, and this is the same thing. It just reiterates all the things that we just talked about. The hepatocytes, they secrete bile. When they secrete bile, they go into those bile ductules of the hepatic portal tria. Before it goes into there, it goes through what structure again? The bile. Hepatocytes makes bile, right? They go into those little vessels called the bile canaliculi. So bile caniculi. Okay, and they go into the bile ducts or bile ductules. That's fine if you just say the bile duct. That's fine. And then from there you have it coming from the right and left hepatic ducts, right? And when they fuse together, it forms the common hepatic duct. Common hepatic duct. And then from there, now you could go into two directions, right? You could either be stored in the stored in the gallbladder. If it's stored in the gallbladder, it has to go into what? Cystic duct, right? But it could also just go out and continue on its pathway. Yeah, but if it goes to the cystic duct, it goes to the gallbladder. And when we start eating fatty foods and stuff like that, from the gallbladder, it goes through the cystic duct again. Okay, and from the cystic duct, it will go into the common bile duct. And when the common bile duct fuses together with the major or main pancreatic duct, it forms the hepatopancreatic ampulla, and this is just the enlarged part. And then I'll go out of the hepatopancreatic sphincter. We'll talk about that in just a second. And goes into the duodenum through the major duodenal papillae. Okay, the pancreas has 3 regions to it. It has the head of it. The head is just basically this part over here. The body is the majority of the pancreas, and the tail is just this very end over here. Its function, and there's 2 functions. There's an endocrine function. The endocrine function we've talked about in the past, right? It releases what hormones did we talk about? What hormones does the pancreas make? When they have if we talk about the pancreatic islets, they have the alpha cells. Alpha cells make not insulin, but glucagon. While the beta cells, they make insulin. And also some somatostatin and stuff. But its exocrine function is for these digestive enzymes and bicarbonate. And we'll talk about, you'll know more about this in the physiology. But yeah, and what produces the exocrine juices again? Do you remember? Where are all the cells around the pancreatic islets? Pancreatic acini. Pancreatic acini. Very good. Okay. Pancreatic islets, once again, is the endocrine function. Okay, so all these are pancreatic islets. And the acini are all these other cells that surround the acini. Surround the pancreatic islets for the exocrine function. So each one of these acinis are called acinar cells. When they make the pancreatic juices, they will go through this main pancreatic duct. This is the main pancreatic duct. The main pancreatic duct produces the bicarbonate. Do you guys remember what bicarbonate does again? It's alkaline, right? It's alkaline, meaning that what kind of pH is it? Very high pH. Very high pH. Why do we want very high pH going into the duodenum again? What else is coming into the duodenum? The acid from, hydrochloric acid from the stomach. What cell makes that hydrochloric acid again? Starts with the P. Parietal cell. The parietal cell of the stomach. Very good. Okay. So this helps neutralize it. That's one function. And number 2, the enzymes that are produced by the pancreas tends to be more active at higher pHs than at lower pHs. Okay. Once again, there's this hepatopancreatic ampulla, which is just this part where it's a little bit more widened, and this is where it merges with the common bile duct that we just talked about here. Okay, and goes through this structure within the duodenum called the, once again, the papilla, right? The major duodenal papilla, and goes through this sphincter here. The sphincter is called the hepatopancreatic sphincter, also called the sphincter of Oddi. And this is made of smooth muscles to help regulate the amount of pancreatic juices and also the bile that is coming through it. Okay. And then it goes through that major duodenal papilla into the duodenum. On the other hand, there is an accessory pancreatic duct, and it goes through the minor duodenal papilla over here. Yeah. Mm-hmm.
The hepatopancreatic sphincter is the top one?
Oh wait, the hepatopancreatic sphincter you can't actually see here, but it's just a smooth muscle that surrounds over here to contract or relaxes depending on whether it wants things to go through it or to prevent the pancreatic juices or bile from going through. Okay. So yeah, you can't actually see that. So for pancreatic juice flow, once again, pancreatic juices, the enzymes are made from, once again, from the pancreatic, not the islets, the cini, right? The islets, they make the, yeah, glucagon and insulin. Yeah, correct. So the cini make the pancreatic enzymes. And the enzymes, they could go through two different ducts, right? One is this one here called the main pancreatic duct. Okay. In the main pancreatic duct, now the ducts themselves, they produce what again?
Pancreatic juice?
No. It will mix. The pancreatic enzymes will mix with. What was the alkaline? Bicarbonate. A bicarbonate, which the pancreatic ducts make. Okay, so this main pancreatic duct makes bicarbonate, goes through here. If we continue with the main pancreatic ducts, it fuses with the, what do you call this structure over here? Common bile duct, okay, and then goes to this enlarged area called the ampulla, right? The hepatopancreatic ampulla, and then goes through this little structure here. This is called the major duodenal papilla. Of course, once again, there's a sphincter in between here, the sphincter of Oddi. And then you have the minor papilla, the accessory pancreatic duct going through the minor duodenal papilla here. Okay, and both of these empty into the duodenum.
So does the minor one also have a sphincter?
No, I don't think there is a sphincter for that one. All right, so that's it for. Let's take a break.